go. We have the last Friday seminar was one of the best seminars I ever attended. I consider it was a perfect seminar. It was very good, very interesting, and very inspiring because it was about moving away from petrochemicals by using biotech and alternative strategies, so trying to find microbes that would be able to generate energy, alternatives to petrochemicals, and all the products we have, everything we're wearing has a lot of petrochemical products in it. So we got and so then this Friday it's going to be a similar one, really pretty interesting stuff. In this room at 9:30 on Friday, like you got nothing else to do, and you have nothing else to do also except after class on Thursday, hang around and practice kicking a ball. Then here's where we're at. So people are done with their term paper. Good job. I now have to grade them, and I'm always complaining I have too much work to do. So the grading of the term papers is going to drag out, and as I finish them individually, I will email you comments and your score grade like that. It's going to go right in through to the end of finals week, right? So we have our final a week from today in here, and I'll still be grading those term papers, and I'll be grading them during the final. It'll just be like that, and all the way till Friday. I expect to get, I always do try to prioritize getting final grades in. I expect to get them in by the Friday of finals week. And so that means I'll have to get all, your grading done by then. So as soon as I get them done, I post them. So, you'll see your grade as soon as, I get it done. And I try to give you, the fairest best grade, and we can follow up on that. I'll have the term paper. You can always, get a hold of that. The final exams, you can always get a hold of yours, one way or the other, and follow up on that. And so that's how we'll finish our course, and we'll talk more about it. The final exam is just the exact same format as the first two exams, so I have plenty of time to finish it. If you don't get here quite at 8 a.m., don't fret, just come in quietly, and again, go to the bathroom, take your phone out, turn it off, put it on a thing in front of you, and we'll get it done. I'm not in any hurry to leave at 10 a.m., so if you could start a little bit late or a little bit more time, it should be good. But, the exam is just the same length and style as our first and second exam, and the questions will be the same, just direct, boom, 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 and no kind of, well, what's the big picture, write about the big picture, just like, what's this, what's that? And we have this new material, that'll be on there, and then all this other stuff. And you can see in the new material, we're still building out from the older material. It's all laced together. And we're just trying to build this fundamental foundation like that. So we'll talk about more new material today and Thursday, but this can also be as much reviewing as you want. I can meet with you or, plan times like that. So that's term paper. Then I kept that answer key video up because a student just followed up. I marked one wrong, so make sure you go through it, because I make mistakes. And also, clarify it, learn from it because, we want to learn that material because it'll be on the final exam, but also the final exam will be that same style of question and answer, stuff like that. And then there's our final exam. So that's where we're at. So we will roll forward here on what we've been talking about with RNA processing and the pathway of gene expression, and just keep talking about that kind of stuff. Again, the book covers the topic somewhat in scattered locations, so you don't need to study your book for this. But it'll be important stuff on our exam from our class. So do people have questions about anything at all? Yo?
Going back over the
material. You want to know about that time my big brother and I found a suitcase full of jewelry in the woods? Sorry, we'll have to talk about that after class. But yeah?
Going over the material about
it was
gene
pretty
protein
cool.
coupled receptors.
It was actually really cool.
I only see you talking about G alpha stimulatory subunits.
Yes.
Do you care about any of the other associated things?
Yeah.
And we only talked about the...
Not for us, for this class, yeah.
For this class.
Yeah.
And you only care about the cyclic AMP pathway. You don't care about downstream calcium or other GPCR associated pathways?
No. Just to keep it focused on, what we covered in class. There can, there could be, there will be, a couple of questions on the exam that are from the book that weren't covered. We care about that book, but you've seen the emphasis. The big emphasis is using our class to find out what's most important in the book. Yep. And then dive into it a little bit further. If you can study more and learn more, dive in, because that's the challenge of being a biologist, is to be able to tackle something like our textbook. Yep. thank you for phrasing all the questions as, do I care? Because I have quick answers to that on a Sunday morning. No, I don't care. We should just be out hiking. Yes, and that's the truth, right? And so, yeah, no, watch me look out the window. Okay, other questions people have about anything at all? And then just interrupt, again, anytime, any point in our course, any topic in our course, it's my job to tell Lisa where we're at. So again, we have this nice visual, this nice physical layout. We're in the nucleus, we're in our classroom, doing all this stuff in our classroom, and then we exit the nucleus through the nuclear pore doorway, and we're in the cytoplasm like that, right? So we're talking about that, and we've been talking about some important steps in this RNA processing. And on the one hand, that processing pathway is pretty routine, pretty automatic. On the other hand, there are points of regulation, and so there are specific examples of specific genes that we've learned about, like MYC, that have things like a microRNA like LET7 that's destabilizing MYC RNA before it leaves the nucleus. So there are these stories in the nucleus about regulation of gene expression in that RNA processing pathway. And so we learned some about the pathway and a little bit about some of the regulation that can occur, right? So this 5' cap is poly A tail, so it has a nice visual. That's what it looks like in the cytoplasm. It's wrapped up like that, and that's how it runs into the ribosome. The 5' cap and the poly A tail. And this is here, but it's a good way to think about how to summarize what we need that's important. 5' cap and poly A tail. They're found on mRNA. So that's coding gene RNA. There are lots of RNAs that are non-coding RNAs. They don't have a 5' cap, and they don't have a 3' poly A tail. So these are coding genes, and there are lots of non-coding RNAs. We'll talk about them a little bit. So these are both true. This is both true for both of those. Both the 5' cap and poly A tail are required to exit the nucleus. They're required for translation by the ribosome in the cytoplasm, and they're required for mRNA stability. And we saw that the poly A tail affects the rate of mRNA degradation. It gets shorter and shorter and shorter. So there is an example of how that's regulated. So it's a nice way to look back at what we learned, and I could do this, but that's the goal of learning, is you organize it in a way that you're like, now I'm ready to have a test, because this is the thing about that, That's what those are there for. Okay? And then we could draw a picture. Again, you don't need to know the chemical structure, but a 5' end and a 3' end, and we talked about the structure of DNA and RNA that leads to that organizational strategy of drawing DNA and RNA all the time. And so 5' cap, then the 5' untranslated region, then the actual coding region, then the 3' untranslated region, and then a poly A tail. And we learned in the 3' untranslated region, that's where the poly A tail gets attached. That's where enzymes recognize it and attach it. We learned in the 5' untranslated region that there can be portions of that affect the regulation of gene expression. So the 5' untranslated region and the 3' untranslated region are important for regulation of gene expression in the RNA after transcription, right? And this is just a nice visual, obviously not in our book, that just shows there's the mRNA in the cytoplasm, and here's the 5' end, the 3' end, it's got that poly A tail. And this shows the mature transcript, and then here's the primary transcript before it gets cut. So before it gets cut and the poly A tail added, in the 3' untranslated region, there are a couple of sites that are recognized by enzymes that cut and add poly tail. Then here's the genomic DNA, or the DNA, that shows that those are in the DNA also, of course. Those sites like that, the 3' untranslated region is part of the DNA also, and that's where that information is. Yep, there could be a mutation in that and make a problem, with that gene. Yep. Okay, great. So we're just reviewing that. We talked about splicing, and we talked, you could draw a picture where there are introns and exons in the DNA, and also the primary transcript, and in the mature RNA, there are no introns. They get removed by splicing, and we drew a picture of this, and you could draw a picture like this. So there's the DNA, it has introns. There's the primary transcript, it has introns. There's the mature RNA as it's leaving the nucleus, it does not have introns. So introns are spliced out in the nucleus as part of RNA processing, and you can draw a map of a protein and show that it's made up of exons, not introns. Yep.
This is a great exon map of a protein. Does this correspond to domains?
So people have wondered for years, why do introns exist? And there's no good reason. Most of the time, the information in an intron is just junk, literally. And people say, well, how do you know it's, you haven't just discovered a reason for it? And it's because people have studied it and it's just junk. But we can't just say introns are junk, because sometimes in an intron, and so let's just go back to this, sometimes in an intron, you could have an enhancer, because enhancers could be upstream of a gene, they could be downstream of a gene, they could be in the transcription unit, because they could be in an intron. And they would work just fine, because remember, the exon, or the enhancer, is just physically tethered to the transcription unit. Doesn't really matter where. So, introns are junk, well, but they could have an enhancer, right? So you have to be careful to say they're junk. Another thing is that introns can be very long, and sometimes they have a whole other gene in one intron, And so you're like, whoops, that's not junk, right? But in general, yeah, introns get spliced out, of the transcript and just get destroyed like that, yep. And let's see, but you asked questions, what was your question again?
It was about...
Oh, about the mapping. So yes, in general, these introns seem to just be random garbage and not very important. They become important especially when we start splicing, because we talk a little bit about this idea of, well, some genes have alternative splicing. And it seems to correlate with the more kind of complicated the organism is, the more likely they have this alternative splicing, and just more and more of it. And some scientists think that's a big way that organisms got more complicated. Remember I said there are just almost the same number of coding genes, protein coding genes in all kinds of different animals, real simple animals, really sophisticated animals. So how did we get sophisticated, And people really want to be careful about describing that, because we aren't, we're not really sophisticated. We do have more complicated bodies and life cycles. And so this alternative splicing is a big deal, and it's a regulated deal. So that's where the introns come up as significant. So then, to your specific question, Sam, so over the years, some people have, when they first found them, they thought, oh, so this is going to show a kind of way of putting a protein together. So for example, we talked about, the estrogen receptor has a DNA binding domain and a ligand binding domain and a transacting domain, right? Well, it would make sense if each one of those would be a exon, and you were able to kind of mix and match the protein product like that, so that the exon would match the domain structure. And for some genes it does, and for many it doesn't, like that. Yeah, it happens to match the domain really nicely for the nuclear receptor gene family. So what I just described is true for the estrogen receptor, is that there is kind of a domain organization to the nuclear receptor gene family. But for lots of genes, the intron locations relative to the final protein product, they don't really make any sense at all. Yeah, okay. The question people have. Okay, so we talked some about splicing, and there's the spliceosome, so that's a term that you should know. There's a protein and RNA complex that splices out the introns. So that would be what's regulated when we talk about alternative splicing. And then we talked for some time about this RNA interference, and there are two types of RNA interference, and one is involved in RNA splicing, and it uses siRNAs, and it involves degrading the RNA in the nucleus, and it's going to turn the gene expression down. The other is after exiting the nucleus, and it's by microRNAs, and they block translation. And so you see in this pathway, this is the RNA processing part, and then we kind of have to take our our RNA interference story out into the cytoplasm as well. And one of the things is, the scientists have known for a long time that there are really kind of these two types of these small RNAs. So that's how they got two names, small interfering RNA and microRNA. And they do have different structures like that, you know, and that's not something we're worried about. We like to think about how they do their inhibition, their knockdown activity, and one primarily by destroying the RNA in the nucleus, and the other primarily by blocking translation in the cytoplasm. Okay, and so you know we have these two categories, and that's what we really wanted to learn about from there. RNA interference is the general term, and it knocks down gene expression. And these little RNAs have targets that they're knocking down. And then I told this at the very end of class. Here are the scientists winning the Nobel Prize for RNA interference. That's what everybody understood. RNA interference, what was discovered, was a natural process, and then scientists quickly realized, well, they could take advantage of it, exploit it in the lab, and design these small RNAs to knock down any gene expression. You know, you could be studying insulin, and you could knock down its expression, and you know, go for it. And so you see, this guy's really surprised when he won the Nobel Prize, because he's like, other people are studying the same thing. And it's one way to kind of appreciate the distinction between these different types of RNA interference, because these early folks were really awarded it for that small interfering RNA and the destruction of RNAs, especially because it was a mechanism to destroy anti-target genes. So it was a natural process. These are natural processes found in both plants and animals, but it was also something you could really exploit and get a lot of value out in a research lab. And then just last year, these guys won a Nobel Prize, and they all studied these different topics in the same species. And it was just startling, and I described about this because I know all these people. It was, at meetings and stuff, and they're the students in their labs and stuff like that. Lots of friends of mine work with these people. Are they all in Massachusetts? I think they all did their work in Massachusetts, yeah? And so, they're like, these people were like, why didn't we win Nobel Prize? We're cooler than those guys, because they literally are cooler. And this is on the World Wide Web. And it was weird, because I don't think anybody, including the Nobel Prize committee, was really understanding what they were doing, but they were really kind of distinguishing these two strategies for RNA interference. One in the nucleus, that was the first one, destroy the RNA. The other in the cytoplasm. The cytoplasm and the microRNAs are far more common in nature. Both are in nature, but the microRNA is very, very common. And so we have examples of both of those in us. Great. Okay. And so then I just want to point out, this is all about coding genes, and we just told a story about coding genes, and we really haven't talked about non-coding genes. They also have RNA processing, and we'll just learn one term for that, and it is, let's get to the term, it's called RNA editing. And it's a type of post-transcriptional regulation. And what it involves is covalent modification of individual nucleotides. And so it's good to look at these non-coding RNAs. This is transfer RNA that is used as part of the translation process. So the amino acid is joined to tRNA and brought to the ribosome for translation. So hopefully you remember tRNA, transfer RNA, and it just shows all the many modifications that occur to tRNA in the nucleus after transcription. So this is just called RNA editing, and it just means modifying RNA after it's transcribed. And it's it's very important biochemically, especially for non-coding genes. So let me just scroll back to show a couple of images about it. All right. So the first one is that these are non-coding RNAs, and you do not need to scramble to write down this list, but here's some famous examples of non-coding RNAs. So transfer RNA, that's involved in translation. Ribosomal RNA, that's involved in translation. We talked about that some in chapter 10 because there's a nuclear body called the nucleolus where the ribosomal RNA gets transcribed and the ribosome subunits get assembled. So we need to know something about that. And then we just talked about microRNAs and small interfering RNAs. And these are just, again, not a term we need to learn. We learned about those some. Not a term we need to learn, but just to mention, they're the RNAs that are in the spliceosome. So lots and lots of non-coding RNAs. Here's just a list of even more like that. I highlighted this one because we'll talk a little bit about it, and we don't need to learn that term, but they're referred to as referred to as non-coding RNAs, and they help regulate transcription. And so there's a lot of science beyond the coding genes. The coding genes, again, are genes that code for proteins, and these are genes that do not code for proteins, but they do important things because the RNA is the product. Here's just a graph from chapter 9 in our book, and it just shows that when you look at the protein coding genes, and this is a log graph, right? So 1,000, 10,000. So there are, some significant differences here, but it shows yeast, it shows that nematode, it shows a plant, it shows a fruit fly, it shows a mouse and a human. You can see, that how can we make a human when it just doesn't have that many more protein coding genes than yeast that causes bread to rise? Yeah, it's really startling, yeah? I'll tell you a quick example of that. So when I was first year grad student, I started to date a second year grad student that I eventually married. And the first year we dated, we went on a cross-country road trip to go all the way from New York to Seattle to meet this scientist who was studying yeast and the cell cycle in yeast. And along the way, we met our families. And we stopped in this town in western Wyoming, and it was a little Mormon farming village. So very traditional religious people who do not believe in evolution for sure. And it was a dairy farm where my girlfriend's paternal grandparents were dairy farmers. And they would have this routine. I grew up in a farm setting, so we just jumped in, helped me with their milk in the morning, milk in the evening. And in between, they chat and they do other chores like that. And every other day, Grandma would be in the kitchen in the afternoon, and she'd be making a couple of loaves of bread, right? And so we haul in there, hotshot grad students talking about, our organisms and talking about, evolution, like that, and stuff like that, really just casually, not even thinking where we're at, right? And these people are like not impressed with us because we're from east of the Mississippi. They literally say that to us, like you're from east of the Mississippi. And we're like, well, we checked a map. We actually crossed it to get here, like that, right? And so in the afternoon, the grandmother, and remember her granddaughter is a yeast, molecular geneticist studying it. We're going all the way to Seattle to meet with, yeast people. I come in, and the grandma is kneading the bread dough, which has this yeast in it. That's why it's rising, right? It has this organism that's yeast that's rising. And as the grandma is kneading the dough, she's chanting under her breath. Her back is to me. She's chanting under her breath, I am not a monkey. I am not a monkey, right? Because she, is just really struggling with this situation, right? Like that. And I just backed out. I'm just backing out of there, because I'm like, that's okay. But just think about it, right? Because it's 2 generations of people, right? Or 3, right? Grandmother, father, daughter. And they go from somebody who says, I'm not a monkey, to granddaughter who's studying the organism she is working with in the kitchen. And the granddaughter would say, well, yeah, you're a monkey and you're also like yeast. It's shockingly similar to yeast, Grandma. And she would just, right? She would just blow up, right? So it's amazing to see this. And people in general, the scientists, when they first sequenced yeast, they're like, yeast has got like 8,000 coding genes. And, we must have 100,000 for sure. And we only have like 20,000, And so what's the deal? And I just want to show you this because over here it shows microRNA coding genes, right? So they're non-coding genes that are focused in on those RNA interfering genes that are microgenes. And you can see that yeast really doesn't have that, and then we see some of it, and we see, considerably more of it as we go kind of toward human, like that. And this is those long non-coding RNA genes that I mentioned. You see a big difference in that, right? So these non-coding genes play a big role in life on Earth and, a big role in our bodies, like that. So again, just one term out of that whole story, and that is, well, 2 terms, right? Coding genes code for proteins and have a coding region. they get a 5' cap, 5' untranslated region, coding region, 3' untranslated region, poly A tail. Non-coding regions non-coding genes, they don't code for RNAs that have any of that. They're different. They do get processed. One of their big processing stories is this RNA editing, modifying the RNA nucleotides after transcription. So a lot of RNA processing of that. And you know, complicated biochemistry and organic chemistry. That being said, just to be aware of this, there are some examples of RNA editing in mRNA, in messenger RNA, where the actual sequence of the RNA nucleotides changes after transcription. And it's unusual, but it is a specific type of regulation that's found even in some coding genes. So, you know, again, we have a good foundation from our course, but as you learn more, you find, oh, there's some of that in mRNA too. Yes, there is. And so for specific coding genes, it's important, you know, essential part of their processing story, RNA editing. Most mRNA genes, none of it, but a very small number, they actually undergo this RNA editing as well. Great. So we did a good job talking about inside the nucleus, and then when we look, right, that's where we get to the exit the nucleus, right? So that's our chapter 10 topic. That's the nuclear pore classroom door. We got to get out of the nucleus. We have the mature mRNA. We have the mature non-coding RNAs and are ready to leave the nucleus if they need to leave the nucleus to do their job. So just to look at that again, remember, this is chapter 10 figure, and I don't think there are any terms from this that we need to learn, just to show you that here it shows, here's mature mRNA leaving the nucleus. There's the 5' cap, there's the 3' poly A tail, right? It needs to have those to leave the nucleus. It doesn't show this in this figure, but it's been spliced, it's had the introns removed. Maybe there's a little bit of RNA editing, probably not, but maybe, and it's ready to leave the nucleus. So it leaves the nucleus, and that's a regulated process. It has to get exported. And you can see it's showing here that these RNAs always have, and it's true for coding RNAs and non-coding RNAs, they always have proteins bound to them. And it's really the proteins that are recognized by the nuclear pore complex and allowed to leave. And you see as they leave, some of the proteins change. And so you can see that that's just part of exiting nucleus. And again, it's not a simple door. You know, that was a big take-home message of the nuclear pore in chapter 10 is when we leave proteins in and out of the nucleus, they need to have these helpers, important to get in and export to get out. And that's true for the mature RNA. It's a regulated process. Yeah, okay? So that just means that, you know, that's a potential important step in gene expression. Yep?
Can you tell us what helicase is doing here?
Yeah, so just in exporting, so a helicase is change the structure of the RNA. And that's one way that some of the proteins are let go. When we think of DNA and RNA, we usually just think of them as like pieces of cooked spaghetti. But each of them actually has a three-dimensional structure and proteins bind to them with specificity. And so what this helicase is doing is it's changing that three-dimensional structure and helping the RNA to shed some proteins and then have somewhat different organization to bind some new proteins. Yep. But again, not definitely not on the exam. So even if you say this is from the book, I'm like nope. But it's good for us to see that again because it's part of the bigger message of the export and import into the nucleus is really a very regulated process, and so potential for control. Yep.
Zooming in on RNA editing,
yes.
You mentioned that RNA editing is actually changing the nucleotide sequence, and so is that similar to the methylation of cytosine, or is it completely removing nucleotide bases and putting in new ones?
That's a good question. And so sometimes and again, kind of beyond the scope of our course, but there are some examples where the coding region codons are being altered by the modifications. And they're covalent modifications that just lead the ribosomes to recognize it, in the process as a different codon. Yep. Yeah. Yeah. And there are other kinds of modifications too that would affect the rate of translation, from the 5' untranslated region, stuff like that. But yeah, there's some cases of RNA editing and messenger RNAs where, and I don't know if that would be in humans or not, but where the actual coding is altered. Yep. Yeah. So, and again, in general, not happening, but when it does happen, it's very specific and it's regulated and it's an important part of that gene's expression pathway is this RNA editing. Okay? So this is again just the nuclear pore showing, well, the RNA's got to get out, and it's really the proteins that are bound to it that are regulating that, so the mature RNA gets out. And again, we don't need to learn more nuke proteins, but here are those import and export reads. So we remember important is the word we learned for getting into the nucleus, and exportant for getting out. And this just shows that there are more of them, and some of them are involved in that RNA export. But we don't need to learn those terms, just to show you that. That's some of the stuff we didn't learn. And here's that, we looked at that slide for the mRNA. Here's some small non-coding RNAs that are regulated in their exit and entrance. So this is just showing that there are some small RNAs that have proteins bound to them, and so they need to first exit the nucleus and get organized with the proteins and then come back in. So again, the nuclear pore and exit and import is just a really kind of a surprisingly complicated gate that you have to get in and out of, and you always need to have help to do that. And that's that. So that just loops us back to chapter 10 a little bit because we want to get out of the nucleus after we finish this RNA processing. So we also want to talk a little bit about what's happening in the cytoplasm, and a lot of that story will be that post-translational regulation. So we've already got a lot of examples of that. You could list a lot of examples because it's all the examples of phosphorylation, it's all the examples of unstable proteins, it's all the examples of localization of the protein. FOXO's stuck in the nucleus. Those are all examples of post-translational regulation and modification. Also remember when estrogen binds to the estrogen receptor, that's an example of post-translational regulation. We're regulating the activity of the protein. And in that way, we're still affecting gene expression, right? Because the gene is the primary target of natural selection and evolution. And so we want to see, well, what's that gene's function and when does the function get used? And in some cases, even the protein is being switched on and off. The gene's activity is, in the case of estrogen, the estrogen receptor gene's activity is dependent on estrogen, right? And so it becomes a long story of, potential regulation in that pathway. So I think maybe I have that. No, I don't. Okay, so that's that. So we should pause there and talk about that a little bit. And last week, I think a week ago, there was a question, and so I kind of jumped ahead into some of these slides about this aging and muscles and aging. And so I just kept those slides in because this is really where we were going to talk about it. And so, that's the stuff we started our course with, is making muscles. And so here's just a slide just to kind of tie into what we just were talking about with the original videos, right? So we started our course and we're like, making muscles and differentiating muscles is all about activating new gene expression, okay? And we got the MyoD transcription factor binding the myogenin enhancer and activating transcription of myogenin, and myogenin goes on to make a protein that's a transcription factor that helps activate muscles. So just a really great general example of differentiation. That's the same way that our brain and our liver and so forth get differentiated. We always have genomic equivalents. We've got all the genes, we're not yet using them. We got the muscle genes, we're not yet using them, so let's start using them so we'll have muscles, right? Same thing with the pancreas. All the cells have the pancreatic genes, not yet using them, so pancreas differentiation is a similar story. And in that early step of making those muscles and pancreas cells, it really is dominated by transcription regulation. When you really dive into the details, you find there are some examples of RNA processing stories in muscle differentiation. So let's just show that for a second and mention that we gave accurate but a really minimum explanation of transcription regulation and muscle differentiation. So I mentioned that before. There are more transcription factors that are involved, but we talked about some really famous ones to get a really nice lifelong model of regulation of gene expression during differentiation in any animal or plant. So here's just some, a recent paper post-transcriptional regulation of myogenic transcription factors during muscle development, like in pathogenesis, right? So nothing for us to learn, just the reality that, yep, there are examples of RNA processing regulating muscle differentiation as well, like that. And so we'll just look at the abstract of it. It says, recent studies have found critical regulation of these myogenic transcription factors. They're talking about MyoD, myogenin, and others at mRNA level, including subcellular localization, stability, and translational regulation. Great. You know, why didn't we talk about that? Oh, just because it's, you know, it's more advanced. It's not the big story. It's not the classic story. It's not the take-home message, you know, but just so you're aware of that. So that happens, okay? And so, you know, the pathway, right? The big focus is muscle differentiation is getting those genes activated, getting the transcription going. And it turns out that, you know, get them done, good. They get to do their work and we make muscle. There are some stories here too, specific examples, right, in normal development and disease where there's some disruption in muscles. Okay, yep. All right, so that's that, just that. Then, it says here too, so changes in post-transcriptional regulators like RNA binding proteins, right? So we learned a little bit of RNA binding protein. MicroRNAs can contribute to muscle wasting. So when we talk about the first part of our course, we talked about making muscles, and we talked a little bit last week about what, you know, when we get old, it's part of the normal aging process that the muscles get thinner and weaker, and people don't have that much capacity to recover from a muscle injury or certain muscle diseases like that. So it turns out when we move to that time, it's just a good example of where some of these post-transcriptional regulation occur. So what we just learned is early, what we talked about in our course, it's about transcription regulation. There are some examples of post-transcriptional regulation and post-translational regulation. But it's, you know, it's mostly about, let's just get these genes turned on. Because what we're doing here is we're saying, let's just naturally move to the other end of the life cycle and just say, here's an example, the same topic, but at this point, yeah, there's still transcriptional regulation, but in aging and diseases associated with aging, turns out there are kind of a lot of examples of RNA processing, RNA stability, post-translational regulation. So that's just a good way to learn about stuff, you know? That both are true, transcription and post-transcriptional regulation at the start of life and toward the end of life. But just the big emphasis there is on transcriptional regulation, the big emphasis here is on post-transcriptional regulation. So it's just kind of satisfying to move your learning around and recognize that we're just moving around to kind of provide a different place to kind of store some introduction to the stories. So what we just talked about with RNA processing, that turns out to be kind of a big story in that aging thing of muscles, right? So it says changes in post-transcriptional regulators like this, those are big story in that aging and diseases associated with aging, you know, like that, okay? Okay, so here's something, and we'll talk about this some this week. Here's the scientists are studying the muscle transcriptome. And what they mean is they're studying just all of the RNAs in a muscle. They're not really focused on transcription. They really want to look at the RNAs and what's happening in RNAs, because in an aging person, there's a lot of changes that are happening at that level. So we'll talk some this week about some methods to be able to study RNAs especially, and they call the total RNAs the transcriptome, everything that gets transcribed. And let's look at all those RNAs. See how they're getting spliced and see what non-coding RNAs are involved in RNA interference and stuff like that. So muscle transcriptome provides the first insight into the dynamics of gene expression with progression of age in sheep. Sweet, right? Sheep are great. Sheep are fascinating. We were sitting literally, oh my God, we were sitting in that farmhouse in the afternoon after doing chores and just leaning back, and Grandpa just says, I find sheep fascinating. And I was like, right on, because they are. And I don't know if any of you have ever hung out with livestock, but individual livestock have as much personality as any cat or dog. And sheep really are fascinating if you get to hang out with them, because they are. They have a different attitude. They're like, we prefer sheep. We really are not your best friend. We like to hang out with sheep. But they have different personalities, and they're just really interesting characters. And so we're just like, right on, Grandpa. Yeah. So sheep are fascinating, and they are fascinating when you do research on them too. Yep. So that's just that example. And then here's another paper, I guess, role of microRNAs and long non-encoded RNAs in sarcopenia. That's this aging of muscle process. Yep. Like that. So just a little overview of this, this post-transcriptional regulation plays a crucial role in sarcopenia, the age-related loss of muscle mass and function. It involves the regulation of mRNA stability, translation, modifications after RNA transcription, impacting muscle protein synthesis and degradation. So again, there's transcription stories too, but it's a lot of that stuff, a lot of stuff that we've been talking about in the last week. Yep. And so here's just a post-translational story, post-translational regulation of muscle growth, muscle aging, and this aging process. And it says numerous post-translational modifications such as phosphorylation, right? So we're talking about modifying the proteins and regulating acetylation, ubiquitination, simulation, glycosylation, glycation, methylation, S-nitrosylation, carbonylation, and S-glutathione are involved in the regulation of muscle health and diseases. And they're especially talking about these older age diseases and states, right? Probably some examples of that in young people and embryos and fetuses and stuff like that, but not such an emphasis. There's just more emphasis in these kind of stories at that later time. So we just put some of our introductory memory in that study, right? So post-transcriptional RNA regulation, post-translational protein regulation, like that. And so that brings up this slide, and Sam asked me where to get that slide, and that's where I got it, from that review. And this just shows in this focus on post-translational regulation in muscle, and they're really thinking about that older person and getting naturally older, and also maybe disease, older, like that. And we're going to learn about, phosphorylation is one of them. We learned a lot about that. We know about that. And then we learned a little bit about ubiquitination, because we jumped to that, and we learned about that proteasome. And that's happening in all of our cells all the time. This review was just focused on old people's muscles, right? So I have this slide about ubiquitination, just to show you again that, ubiquitination plays a critical role in protein degradation. And proteins get ubiquitinated, and that's a tag to recruit them, direct them to the proteasome trash can. They go in there and they get degraded to amino acids. And that's generally the way proteins get degraded. For really unstable proteins like MYC and so forth, there's additional mechanisms. But, that's the good one. And so we should know that. Post-translational regulation is ubiquitination and degradation. And that, we got to make the protein, and we're going to degrade it, because there's a constant turnover of proteins in a cell, and it's a dynamic equilibrium, right? We could get less protein by either decreasing transcription or increasing degradation, and that happens. So that's a post-translational modification and regulation. And so here's just an image of that idea happening. So the protein gets ubiquitinated, and it goes into that. It's a barrel-shaped subcellular organelle called a proteasome, and there are lots of proteasomes hanging out in the cytoplasm and nucleus, and just gets degraded to amino acids, okay? So then let's see what this is. It says, and it's just talking about MYC, and MYC goes into the trash can that way, but MYC is really unstable, so there are other mechanisms that get MYC destroyed. Not surprising. And it's a big part of the MYC story. We're just going to try to, we're trying to keep minimal MYC expression, because it's this super dangerous thing. Okay, and then also beta-catenin, had that story, it gets ubiquitinated, it goes in the trash can, and Wnt is going to stabilize it from that destruction. And it goes in the trash can, but then there are some other mechanisms to get it degraded. P53, another one, right? Because we have good examples of protein degradation in our course and having it regulated, okay? So that's phosphorylation, ubiquitination, and then we're just gonna learn this one other one. There are several, and it's called summoylation, and it's a funny word, S-U-M-O-ylation, and it stands for small ubiquitin, I think modifying. And what you want to remember is it's just going to be a covalent molecule, and it's a little protein. So summoylation is a little protein, and it gets actually covalently added to the protein. So we'll learn those three, right? Post-translational modifications, there are a lot. Phosphorylation, ubiquitination, and summoylation. And it's pretty interesting because, we see ubiquitination, that almost always means we're going to destroy it. We know phosphorylation means you can turn it on, turn it off, right? Here's the summoylation. Here's a summary. Summoylation is a post-translational modification involved in various cellular processes, such as nuclear cytosolic transport. So one way of getting out of the nucleus is to get summoylated. Transcriptional regulation. So one way of regulating transcription is to summoylate the transcription factor. Apoptosis, right? We learned about that. Protein stability. Response to stress. Progression through the cell cycle. So, right, just about every topic we talked about, we could find an example of a protein that gets summoylated, and that's helping regulate that story. And we won't learn of any of them. We'll just learn the idea of summoylation is a covalent post-translational modification of a specific protein to affect its activity. So we have those three modifications: phosphorylation, ubiquitination, and summoylation. Yep. And we don't have any specific proteins to name, just to go there and show that idea, right? That we have those. And you can see there are a bunch of others that, are part of the story, right? And, for a specific protein, like then probably some of your term papers and the receptors, you probably found they were modified in one way or the other, affecting their activity, like that, okay? And so just a little general thing about it. I'll just read through this again, because, we're not learning this in a specific way. But summoylation, the covalent attachment of sumo proteins to specific lysine residues on proteins, target proteins, is a post-translational modification that significantly affects protein function, stability, and interactions. This process modulates protein behavior by altering its localization, its activity, its interaction with other molecules. So summoylation sounds like such a bizarre, weird term, right? Because it sounds a little bit like we're going to wrestle, and but it's actually really involved in specific protein function and activity, right? Here's more detailed. So protein function. Summoylation can change how proteins interact with other molecules, including DNA, RNA, other proteins, alter protein activity, such as the transcription factor activity or enzymatic activity, right? So it's just right through the main part of all of our core. Every day in our course, I could have come in and said, well, here's another example of summoylation, because I happen to think it's really cool. Most proteins are not summoylated, but the ones that are, it's part of their regulation. So protein localization, right? We learned something about that. Protein stability, protein-protein interaction, regulation of gene expression, right? It's just all over the place, lots of examples of that. Okay, so that's that. So just a little bit about this post-translational regulation of gene expression. Okay, so then we'll be done talking about that. So we should pause here and say, oh, our course is done, but no, I still have to get paid more. So we're going to keep going, but let's see if people have questions about anything at all. So let me just look, because I think the next slide is, so this is just a little bit more, so that's really chunking it through, and then this is just a little bit more follow-up on that aging. And we talked about this last week, and I just wanted to go back over it, because we kind of jumped into it a little bit out of context. So again, it's kind of, you have to admit, isn't this kind of satisfying, and don't you think I'm a great professor? Because we started our course with making muscles, right, and we're ending our course with aged muscles and muscle aging and the other end of the muscle story, right? So there's a really important hormone, myostatin. And so it's one of these molecules that goes between cells and binds to a receptor. And the receptor is on the muscle satellite cells and also muscle cells. And so we can talk about that. You know, we talked about other examples, right? We talked about the skin cells under a wound and how PGF is a signaling molecule. Well, this is myostatin is a signaling molecule, and it's a receptor, and it's going to affect the cells, and it's going to have the genomic effects and non-genomic effects. It's just going to be a response. And myostatin is involved in this old age, aging of muscle story. And myostatin, I don't know very much about it, because its story is really curious. It's made primarily, I think, primarily by the muscles. And so I don't know how it's regulated, like how, you know, what's going on with its, why is it there? You know, I just don't know too much about it. But the bottom line is it's a hormone, and it's going to be regulating this natural aging process, and it's affected in some disease. There are diseases that are muscle wasting diseases, where a person loses muscle mass before they should. As a young person, they start losing muscle mass, right? So here's a myostatin negative regulates satellite stem cell activation, right, and self-renewal. So we've said so far in our course, oh, you're going to have these muscle satellite stem cells your whole life, because they divide asymmetrically, and so when you do muscle, they'll divide, and they'll make the precursor myoblast, but they'll also be self-renewing, and you'll have them your whole life. So here we add, at the end of our course, nope. They start to get shut down by this myostatin in old age, and less responsive to the needs of recovering from a muscle injury or, you know, maintaining building muscle mass. So satellite stem cells are quiescent muscle stem cells that propose postnatal growth and repair. Here we show that myostatin, a TGF-beta family member, signals satellite cell quiescence. And so we talked about that last week, that it actually that the receptor is going to activate a transcriptional response that's going to activate expression of that P21, and just say, and I don't care if there's any DNA damage or anything, I just don't want you to divide. So the stem cell, that muscle stem cell, is blocked. And let's just check this. Cell cycle analysis confirms that myostatin upregulates P21. You know, so we said, well, you know, how does the signaling pathway work? And I mentioned that this was one of the receptors that's it's not a receptor tyrosine kinase, it's not a G protein coupled receptor. And at the end of chapter 16, we learned a little bit about some very important other types of receptors like Notch and Wnt signaling. And this is another one. And all we learned about it is that this would be myostatin, this would be its receptor, and it's going to work by activating these transcription factors called SMADs. They get phosphorylated, and they activate transcription. So we said last week, like, for example, it could activate transcription of P21. And it's not P53, it's this other transcription factor that can activate P21 and just block the cell cycle. And, you know, you just go shrug. I'm just like, that's a bummer, right? For the grandpa that thinks sheep are fascinating, and he's sitting there with his myostatin shutting down his stem cells, and he's got to keep working, right? Because he's got to milk some cows, you know, twice a day like that. So it's just a really curious thing, right? And but we understand the pathway, right? We can dive into this literature and be like, oh wow, I can't believe this is going on, right? So we learned about SNADs a little bit, so we can add to that, right? So I just wanted to go to this too, because we got this myostatin. And myostatin also affects this differentiation. So it says that in this pathway, we're going to affect the downregulating the expression of MyoD, for example, and also just saying, you know, yeah, we're shutting this down, you know, through these pathways that we're have some familiarity with, that, like that, right? So just kind of looking at that. And so again, you know, early in our course, we really emphasized this. Now we're just bringing up, you know, these are important in different ways for a lot of gene expression. Yep. So I think that's that, and I think now, yeah, we jump to the transcriptome and that kind of story. So let's pause here and do a concept chat. And so again, if there's somebody that you can talk to yet this semester, say hi to them and see what they think was important things, 3 important things that we talked about today.
myostatin is great with cement pathway.
So RNA editing was one of the new things we did, and it's a type of post-translational modification. And then so it obviously modifies the RNA after it's been but this is actually question of whether or not it was methylation like that, or if it's actually changing the nucleotide, and I didn't understand the answer. So I thought this was so I'm not sure. Yes. So then we talked about utilization was that was like the little protein market, and like more market is more like ACI and like recycling the that's the right word.
Simulation is proved it's like exactly like postology or something.
So what is that do? It just basically changes the post.
I think it's like like I wrote like all things.
Okay, cool.
Like we know this, but no examples.
Okay. And then the hormone myostatin is basically what like cause like protein degradation for old people, which like if it means that it was pretty used and elusive, like that old man could lose his whole life, but he's still right. Okay, well like I'm that.
Yeah.
But no, yeah, I was like, I was thinking about doll.
Wait, remember you said you read the chapters?
I did. I read all the books and stuff just right up my math.
And we'll just do this for another minute.
I'm like redoing all my notes that I wrote, so I'm like,
Okay, and so Juliet, what is one thing your group talked about that you thought was important so far?
We talked about RNA editing for the non-coding genes.
Yep, RNA editing. So, because probably all of you can remember tRNA, transfer RNA from IntraBio, and it undergoes lots of RNA editing. And that is actually really common for those non-coding genes because it's that RNA is going to be the functional product of the gene. And so over evolutionary time, there's been lots of modifications that have evolved that are possible to modify the RNA to make it more of a structural, functional, active molecule. And and it turns out there's a little bit of RNA editing in some specific coding genes, which is surprising and intriguing, and always going to be how important is that? And in some cases, it's critically important like that. Okay, and Matt, what's another thing that your group talked about?
we talked about myostatin hormone and how it degrades muscles in old people.
Yeah, and I would say it doesn't really degrade them necessarily, but it just kind of limits their capacity to maintain healthy muscles. Yeah, and they actually call that this normal aging-related, like muscle mass loss, sarcopenia, and they refer to it as a disease, even though that is just a natural aging process. So I don't really think of that as a disease, but they call it's a disease. Sarcopenia is this muscle disease, and then there are lots of examples, or several examples of these real disease states, like muscular dystrophy and so forth. Yeah, Alex, do you have a question?
Yeah. So just for clarification, so for the myostatin, it makes it so that the satellite cells are stuck in the cell cycle, so then the muscles can't really recover from injury. Is that one of the ways?
Yeah, and then it also can bind to myoblasts and block MyoD expression and activation of differentiation. Yep.
That's the main reason for sarcopenia, the main cause behind that, just the increased myostatin.
Oh, sorry. And I should say I have my phone on because I'm getting a delivery from a big heavy truck transport thing, and they have my phone number, so I just kind of have to be aware of that. And that's what I was doing the other week too, when I was pretending to be looking at my phone when I was supposed to be talking to class. But I kind of have to keep an eye on that. But anyway, sorry, yes. What? Now,
So is the main
well,
is the main mechanism of sarcopenia just the myostatin levels that are
I don't know.
increased in age?
Yeah, I don't know. Is it all about myostatin? Yeah, that's a really good question, and I don't know. Yeah, I don't know too much about that muscle aging story. Yeah. And then let's see. So Cam, what's another thing that your group talked about?
Well, we were talking about a bunch of different things, but one thing that I have a question about
Yep.
is for RNA splicing. So protein families, are they made by the different conformations from RNA, like RNA splicing?
Yeah, that's a good question, and we should go back to that, because remember, I did try to emphasize that. So we learned a lot in our course about, well, there could be a family of genes, and that's really common, so that it is caused by random gene duplication, a type of mutation where a gene can get duplicated, and then it's possible that it could that one or both of those could get some mutations, and so that they could have somewhat different functions. And I always describe a gene family as like a set of screwdrivers, and there are some different types of screwdrivers, and they're all in the screwdriver family, but they're actually different screwdrivers made by different genes. So we have different genes. And here's a good example of that. So we have a protein in our body called insulin. There's an insulin gene, and insulin is a member of a gene family. The insulin gene family includes 3 genes in us, like that. And one of them is insulin, and then there are these 2 other genes, and they're not insulin, but they're very similar to insulin, and they actually have overlapping activities. They're all hormones, they all bind to receptors, and they have some overlapping binding to the receptors. The insulin receptor is also a member of a gene family. So it's all about doing something that's pretty similar. Remember, though, that even though it can be similar, we learned about the BCL2 gene family, and we said, let's take it easy here, because some of those family members are pro-apoptotic and some are anti-apoptotic. So they can have some different, different activities like that. But that's a gene family, and the gene family members are called paralogs. And we saw that with the caspases. They're numbered. Those are different genes. We saw that with the cyclins. Those family members are given letters. Those are different genes. Yep. So now, yep.
Do the different conformations, do people pay attention to those and put them in a group?
So now we're just going to shift to this idea of here's one gene. There's one gene, and it can be alternatively spliced. But it's just one gene, and it can be alternatively spliced, and it could make somewhat different protein products. They're from the same gene, so that the parts of the protein will have identical amino acid sequence, and then parts will just be different. Kind of a modular mix and match, like that childhood game where you put a different head on the organism, the person, and looks like a farmer, and another looks like a ballerina, and you're like, oh, like that, right? So a little bit of a mix and match, and it's regulated, and so that's just one gene. And I think you're asking especially about those, and those different versions are called isoforms. And they're given names. We haven't learned any of the names of protein isoforms, but they'll have maybe a Greek letter or some kind of designation like that to distinguish between these different protein isoforms. And really importantly, protein isoforms are made by alternative splicing of the same gene. Yeah?
Is that like estrogen receptor alpha and beta? Those
So that's a good question, and that's a little bit challenging because there are examples where that lettering is not, the lettering naming system is not perfect. So in some cases, they're actually referring to 2, like in that case, 2 different paralogs. And then in other cases, for example, the estrogen receptor also makes a variety of different isoforms. And so, of course, because the lettering and stuff is not universal, it can be confusing. But for us and for our final exam, we just need to be able to distinguish, right? When we're talking about a gene family like caspases, and again, we didn't need to learn the caspase numbers, but I could say, give you numbers, are caspase 4 and 5 paralogs or isoforms? And they're paralogs. There are two different genes. And I don't think we learned any isoforms. So that kind of helps your answer for the final exam if that question comes up. But it's just an important distinction. And both of them are legitimate ways of making very similar proteins that could do a somewhat different function. And we see a lot of both in, again, and I don't want to overemphasize this, you know, the more complicated organisms like us, we have a lot of gene families. And we also have a lot of alternative splicing of individual genes, right? So it's a way of, like in a toolkit, it's a way of having a more complicated toolkit, right? By your, like, you know, the intro toolkit has got just two screwdrivers, the standard screwdriver and the Phillips. But there are different sizes with different lengths of the shaft and different widths of the blade, you know, and it gets really complicated. You need 40 screwdrivers if you're really fancy, you know, like that. Yep. So does that help clarify? Because I kind of interrupted your question because I thought you were kind of driving at that comparison.
It makes sense.
Yeah, and we just need to be careful to have that foundational understanding of those two different situations. Yep. So other questions people have, we have more time, we're going to kind of introduce this kind of last topic, we'll look at that a little bit, but we can answer any question, you know, because we're doing great. Yep.
We have talked a lot about post-translational regulation of proteins. And one of the things we've talked about is post-translational modification, so covalent modifications of proteins. And I'm wondering if all covalent modifications of proteins are what we would consider regulatory. So I'm thinking of like cell surface proteins that have like are glycosylated as an inherent part of their like synthesis. Yeah. Would we call that a post-translational modification? Like,
yes.
Would we call that a regulation though?
No, not necessarily. Yeah, it might not be a regulated step, but so yeah. So again, we have this term post-translational modification, and that means a covalent modification to the protein, and then a broader term, post-translational regulation. And so post-translational regulation, it could just be like where it's localized, you know, oh, you're not letting it in the nucleus. So, yeah. And and there are lots and lots of post-translational modifications of proteins, and they, I don't think, would all be considered at all regulation. Yep. Yeah. Other questions people have about anything at all? Because we have, I think, 5 minutes left, or 4, or so. Yep.
We talked today about the distinction between RNA editing and RNA processing for coding genes.
Yep.
And I understand the mechanistic differences between those two, but I'm wondering if there's like a useful, like, coming back to the, almost like similar to the question I just asked, like, how can we think of these conceptually as distinct or different?
Well, so first of all, we talked about RNA processing of coding genes. RNA editing is a type of RNA processing. So RNA processing just means post-transcriptional regulation of the RNA by altering the RNA, processing it, altering it. And so we had the classic examples for coding region mRNA of 5' cap, poly A tail, splicing, and RNA interference. And there are more examples of that than just those, but those are the bulk of these post-transcriptional regulation of mRNAs for coding genes. And then for non-coding genes, we'd say they don't have a 5' cap or a poly A tail. They have very little splicing. I don't know, maybe none. And they aren't involved, aren't the targets of RNA interference. But they are there's a lot of processing because they go through a lot of RNA editing. And the way to think about that is, well, there would be, you know, you're saying this is the product, the RNA is the product. And so there would be a lot of natural selection opportunity to make a modification to the product to improve it or alter its activity. So there's just a lot of that. And these are these non-coding genes. And so we said there are a lot more non-coding genes in us than there are in a fruit fly or a yeast. You know, that's one way to make a complicated organism. And that includes a lot more of this RNA editing of those non-coding genes. So there's a lot of that story. But they're both would be both called RNA processing. And the way really to distinguish them is to go back and say, let's first learn about the coding genes and that pathway toward, the product of this gene is ultimately going to be a protein. So on our way to a protein, we got to make this mRNA. And, how is that processed and how is it regulated? And then alternatively, well, it's a non-coding gene. So that's going to be the product. So how is it going to get ready to be its final product and do its function? So like transfer RNA, it just has to go through all kinds of RNA editing before it's ready to do its job in the process of, translation. Yep. Yeah. Yeah, and we're trying to kind of build this framework to organize knowledge and say, oh, we're way over here. It's a non-coding gene. So yes, I expect to learn about RNA editing because it's a non-coding gene. That's going to be the product of the gene. So I won't be surprised that it's kind of a lot of RNA editing occurs in it. Yep. The questions people have about anything at all? So let me just show you this and show you that. So here, we see this thing, and we're going to talk about DNA and RNA and proteins. And so the thing we'll talk about on Thursday is, again, you don't have to look at it, but scientists have really lots of experimental methods to study DNA and RNA and proteins. And now those methods include studying all of them. So in our course, we've gone through and we've talked about MIC, and that's been the tradition of biology. Well, this research lab is working on MIC, and that research lab is working on insulin. And now there are lots of labs that just study the whole thing, and we saw a little hint of that where they saw the muscle transcriptome, and they say, this lab studies muscles, and we just study all the RNAs. We're interested in all of them. So we're just going to talk a little bit about some of those tools, because that's the shape of modern biology. It is really big data sets, because methods are present now to study all the DNA, right? So human genome, it started with that, We should study all the DNA in a human, and it's a lot, but let's just get it all, and then, people can study what they want. So that same attitude is true for the RNAs, because what about them? What are they doing? And proteins, what are all the translational modifications in an aging person? Let's look at them all. Yeah, so we'll take a look at some of that, and there'll be a little bit of learning, a couple points on the exam, but we won't need to look at our book. Our book covers that a lot in chapter 9, so I have some figures from that. But yeah, we're doing great, and it's it's important, but it's extra. If we have Thursday, all the questions you have, bring them on Thursday. Right on. Yeah, we're doing great.